Hello, I'm going to talk really fast to show you how to do the enzyme lab if you don't know how to get started. This lab is in your binder and it starts on page 39. Oh, maybe that's 38. Okay, let's go to the whole purpose of it. The purpose of this lab was to observe an enzyme called catalase work on hydrogen peroxide. Catalase helps hydrogen peroxide break down into two parts, the products water and oxygen. I've highlighted the main point of it and I crossed out parts we didn't do. We didn't do anything about the enzyme concentration. We always were using 3 milliliters of potato stuff, and that has the enzyme in it. And we didn't do any, environmental factors, that is the temperature, the pH. So that's redundant. So definitely make sure you have the pre-lab questions answered like we did in class. Next page, page 39. On this page, you should have your lab data in here. You and your lab partner recorded this. Now you need to go to the Google Sheet. The link is in Google Classroom, and many of you already went to it to put your number in so it could get the class average. So here we have class average for bubble height on top, and then class average for that ranking of the reaction rate 0 to 5 over there. Same thing on the next page. This one has to do with the effective temperature. So we have a little bit more to fill out because of hot and cold. And your data goes here. And only thing you have to fill in for class data is the averages. Go to the Google Sheet, scroll a little bit to the bottom. Maybe it's about row 50. Now for the graphing part, it is hugely important. Don't skip it. And it's going to take you the most time. Leave some time for this. And don't just go by this video. Make sure you read and you think as you make these two graphs. The graph paper is there for you on page 43 and 44. I'm expecting 2 bar graphs, one on each page. The first bar graph is going to be for temperature. And on this graph, you are going to make sure that you include the hot treatment, the cold treatment, and the room temperature or medium treatment. You probably wrote down the temperatures somewhere in your binder, and that would be better than saying hot, medium, and cold. And then also on the other graph, we are comparing pH to the control. So the control goes in the middle, or you could call it a pH of 7. We never actually measured it. And so we'll just have to say a low pH or something acidic. Our control group, we did not change anything about the pH, and then we added sodium hydroxide to make it more basic or a higher pH. You're going to want to color code your bars because one of the bars is going to be about bubble height. So for these 3, the way I kind of sketched it out, how I think it's going to look, is that those 3 are for the bubble height. And so you'll need to have a scale on one side of the y-axis for bubble height. Give it some kind of a color or pattern there, so I know those are all for bubble height. And then you need to have some other bars, all the same color, and that one will be for the ranking, the 0 to 5 ranking, so you just need some kind of a scale on the other side of the y-axis that just brings you up to 5. So this axis here is vertical for bubble height in centimeters. And this one over here is also vertical, so I'm going to use the other side, and it's going to be ranking 0 to 5. If you find that bubble height is easier to graph, easier to lay out the scale in millimeters instead of centimeters, that's quite all right, but you need to do that conversion yourself. So I'm expecting 2 graphs. They will be bar graphs. There will be no lines on these graphs, no connect the dots. And then use this as a checklist down here. You have to have a title. You have to have labeled axes with units. And that says the appropriate scale. Your scale can't jump all over the place. If you're going to go 0, 2, 4, 6, 8, 10, you have to stick with it. You can't go 0, 2, 4, 6, 25, 26, 27 to make something fit. You have to pick a scale that fits. Talk to me if you don't know what that means. Email me. The graph paper I was talking about is here on 43 and 44. You could do it portrait or landscape, whatever works best for you. Count it out before you start using markers or anything on it. And then finally, another big part of the lab is the discussion or the analysis. This discussion or analysis should be written in complete sentences. It does not need to be written for English class. Like, you don't have to restate the question, but you have to write something more than 3 words. Now, we probably need to talk a little bit about CER, claim, evidence, and reasoning. This was a struggle last lab report. So let me write down a few tips about that. A claim is the same thing as a thesis statement in history or English, or for this, it's our conclusion. What did you see? Did you see that high pH gave a faster reaction rate? Or did you see that high temperature gave a faster reaction rate? Make a solid statement. I think higher temperature increases the reaction rate of catalase on hydrogen peroxide. Make a strong first-person statement. You need to probably make a statement, one about temperature and another about pH. When we talk about evidence, I mean your evidence is your data. You have to tell me something specific that you see on your graph. So you use numbers, or you use a slope if you had a line graph, or you talk about a trend in the graph. So you have to back up this conclusion with evidence. It's as if I said, prove it to me. And then you'd say, oh, I'll prove it. Here, look at this data. There's a big difference between a ranking of 1 and a ranking of 5 when we're talking about low pH or high pH. I'm totally just making up that data. And reasoning is not any research on your part. It's not any guessing on your part. Reasoning means connect the 2 things you just talked about. Connect your claim to the evidence you just provided. This is really about your thoughts, why you think you're right. It's a little bit hard to do at this high school level, but it is certainly not you saying that you think catalase is more natural in potatoes than in fern plants. You don't know that, and you don't need to look that up. Your reasoning is just connecting the 2 things you've already told me about. So make this good. Let's try it again. Let's not talk about errors or modifications. Let's just do the claim, the evidence, and the reasoning. Email me if